My background is in genetics and molecular biology. I have very limited familiarity with social science concepts beyond my collaboration, so please go easy on me, particularly since I'll be telling you about network analysis where actually the experts are from social sciences. Network analysis and the maths driving that has very much come from the social study of this, and we're very much as biologists picking up on this. Please treat me nice and easy with that side of it. So I'm going to cover a basic understanding of what transcriptomics is. I'm going to look at some a top-line set of ideas as to what analytical approaches we use. I'm going to sideline into network analysis as one of these, and then I'm going to give you an awareness of some of the archive data that is available that can be used. Should perhaps start by running through this slide and saying we're trying today to follow the order of flow from the hard information that's stored on your genes through to the processing of that via the message, the transcriptome, the proteins that are then coded, and the metabolites that come from that. And of course we have within this flow an increasing interaction with the environment. So what you have with the DNA, we talk about single nucleotide polymorphism. You have things that are fixed, things that you're born with, things that are in every cell in your body. Whereas when we move through to levels to which a gene is expressed, this may change depending on what you've eaten, what time of day it is, all sorts of factors. But I also think that's where the interaction between social sciences and biology comes, because it's these interactions that actually hit the debate between gene, environment, what's most important, how does environment affect genetic basis, how does genetics affect the environment. So I'm going to cover this again. Chris has already looked at this. This is the central paradigm of molecular biology, some people term it. And I'd like to make sure everybody's got this one straight in their mind, that DNA moves to making proteins via genes, and there's an intermediate step called messenger RNA. DNA sits in the nucleus and it never leaves the nucleus, whereas proteins are made and act in the cytoplasm and outside the cell. So the DNA produces a message, a transcript. The messenger RNA leaves the nucleus and it then docks with a ribosome to make a protein. And the three-letter code that was described with the talks on genetics helps lock in the amino acids to the ribosome to form the protein. Now, this is a central part of understanding. DNA, of course, is there to keep all the information and to provide the replication. It divides and allows the generational transition of the information that's encoded in a cell. Transcription allows that to actually work. The gene has to do something. It transcribes itself, and that then is translated into protein. And it's the protein that generates effect. It builds structures, you have enzymes that catalyze reactions, and effect comes from, function comes from the protein. So DNA is the information, RNA represents the flow of that information. How quickly, how much you actually turn the engine to produce some form of effect, and the protein itself represents that effect. Now, of course, a very important point has already been raised with the epigenetics, and that's the fact that this process of transcription varies enormously depending on cell and type. And this is a very key point that we have to address within the type of work we do and understand. There are certain commonalities that we can come back to, but it's the very nature of what makes different cells and how we respond in different ways is represented at this point. So we're starting to look at the interface where genes interact with the environment. So we can have a two-way flow, obviously, between the gene and the environment, but the genes and your basic coding is staying fixed, but that's being affected via the epigenetic pathways we heard about, and obviously we're transcribing our genes to help us affect the environment. There is a second factor I want to bring in here, and this is development. Not only do we have the complexity of the environment, we have the complexity of different cell types, but actually these may all change depending on how old you are. Through the program, through being born, going through childhood, the rates of growth, the trajectories of growth are different, and going through the phases through to old age. Transcription will change, and it will change in every tissue in the body to some level. And we have to start to introduce analytical processes that allow us to factor this in to our study of the information. So the transcriptome is the set of all messenger RNA molecules. That's it. We have really two main ways of actually analysing that at this moment. We have microarray technology and a process called RNA-seq. This is a microarray here. It's a chip which has different bits of complementary DNA on it, which allows it to bind to a reverse transcribed RNA, and we can quantify that and get a signal. I'm not going to talk too much about the actual methods within this. What you need to know is it actually produces a list. And actually, unlike some of the other attendees we can hear about today, the technology behind generating the transcriptome is really fairly robust now. It certainly wasn't when it first came out. If you went back, you can go back perhaps almost 20 years to some of the first chips. Some of the first publications on transcriptomic data were quite disastrous. They were really so badly wrong that they had to stop and actually bring together proper quality control standards. This has all been done, and this is now really quite robust. Being able to measure these levels of transcriptomic action, levels of transcript within the cell, is fairly straightforward. The newer approach is RNA-seq. That's relying on the next generation sequencing machines that are now available. It has its own quality control issues, but again, these are fairly strongly under control, and it's very easy to get data. To give you a rough price for this, you're probably looking at about £200 to £400, depending on who you speak to do one person or one sample from one individual to measure all the transcripts. And that would be with either technique. So we get a list. That list may look something like, or the top of that list, something like we have in the middle of the screen here. This, of course, suffers from the issues of big data. If we have several thousand of these out of 23,000 genes in the human genome, if we look at it at an exon level, we measure every element of the gene, and whether that's transcribed, we might go up to 100,000. We're still in a much easier zone within transcriptomics than they have with genetics. With SNPs, we're talking millions. You're on at least another factor of 10. So actually the issues with false discovery of genetics are really severe. Here, they're only a bit severe. So we can look at this with simple standard statistics. You can apply a simple t-test across the data once you're confident that you have good quality data, or ANOVA, and you can look at p-values and false discovery rate corrected p-values. But this has this issue of generating a list and a cutoff. What does it mean if I've got a gene that I'm very, very confident has changed between one set and another? Is that gene really functional? Is that the one that's really going to do something? Is that the one? What am I interested in the data? And usually I suggest people are interested in some level of functional observation. They want to know why these changes occur. And between whatever two groups you're comparing, they want to know something about the mechanism. And very often it's actually not these things that change really severely that we can measure easily that are the things that do make the change. It's actually much more subtle changes downstream, or upstream in this case. I think it's like the example of, from a systems biology approach, it's like starting an avalanche by throwing a small pebble up a mountain. At that point it's a very small action, but down the bottom of the mountain you've got a huge force of nature rushing downhill, destroying everything in its path. So when you have big changes in a gene expression in relation to something you're looking at, it's more likely to be the downstream consequences of what you're seeing, not the thing that has actually initiated that process. So why do we look at these data sets? I think there are three main reasons. There's discovery. Being able to say, I've got two, say, groups of patients. Can I differentiate these patient groups based on a gene expression profile? Can I look at subtypes within that? There's been a lot of work recently showing within cancers molecular subtypes of that which have come from omic data. Can I use this to compare individuals in, say, response? When we're looking at therapeutic response, very often we have good and poor responders. And it's perhaps the similar idea. We want to be able to actually differentiate what constitutes the phenotype of a good responder or a poor responder. But finally, can we actually use the information we get to enhance prediction? Can we build some form of classifier, a gene signature, and therefore predict response? I think this is a bit of a holy grail, because whilst everybody writing papers on this wants to talk about this, very few people can actually do prediction, and certainly robustly. But it's certainly something that, with more modern approaches in building classifiers, is possible. So how would we approach, or how would I approach, perhaps specifically, a transcriptomic data set? I would to look at the sort of questions I've posed surrounding the comparisons between the different phenotypic groups I would have within patients. And we've already heard about how important it is to define and classify your phenotype. But the first thing I would do would be to come into a principal component analysis type of environment, where I'd see whether, very simply, the groupings coalesce in PCA space. So what we would have, say here in this example of principal components showing the first three components in 3 dimensions, if we colour it up by a particular phenotype that we're aware of, we can start to see that these things, of course, should coalesce to some level. In fact, we might see a cluster that we have no idea what the phenotype is, and that becomes interesting. That becomes something that obviously exists as a group, but we don't necessarily know why. And we can start to actually map back on the phenotype data to try and find out. This here is another example of a type of principal component analysis, where all we've done is we've built out an ellipsoid to sort of map the surface of confidence around a cluster of points in space. Principal component analysis can come under some criticism for essentially being still being driven by sort of fairly linear concepts, and also limiting the amount of variation we can cover when we just say look at 3 components. We can move through things like an isomapping approach, where we can compress the variation, more of the variation, into our ability to visualize it. And we can, if we're concerned about things like collinearity between variables, we can look at approaches like orthogonal partial least square, but they will have a very similar output to what you're seeing on screen here. It allows us to look at the data, analyse it really quite visually, and say, which bits of this, how are they clustering? Is something here not clustering in a way that we would expect with the phenotype that we're looking at? The other way would be looking at a more sort of supervised approach, where perhaps we bring a standard statistical type of analysis to this. There are a lot of description approaches. If you use R, there's a number of different ways within R in which you can actually tease apart 2 data sets, 2 transcriptomic data sets, and some fairly robust ways of doing this. By and large, though, it should be remembered that an ANOVA is actually really quite resistant to shifts in its probability distribution, or shifts in the distribution. And actually, an ANOVA can be quite tolerant of, in a situation where people might be trying to use some complex Bayesian whatever. Actually, you throw an ANOVA over it, you can actually get the same quality of data. Please don't all attack me on that one, but it actually, from the point of view of what we're going to look at with networks, seems times to still be a surprisingly effective technique. If we do this, we can start to examine the data in the way I present here. This is a heat map. And I'll just spend a minute or two telling you what's actually going on here. What you have vertically is each one of these rows would be a gene. And each one of the columns would be a patient or a person involved in whatever phenotype dissection we're looking at or comparison we're looking at. And what you see here is clusters of red. Red means it's upregulated. There's more of the gene transcript in that person than there are in the other people in the sample. And you can see that we have clusters arise, and these clusters, of course, should map, like we saw with the principal component analysis, to particular phenotypes. Or, turning that around, if we see a cluster where we're not aware of that, well, something's going on. This is a molecular phenotype. It's defining some response, and we would like to understand what particular phenotype in the patient that is actually representing. When we find these types of clusters, we start to have the basis of a classifier, and that pulls us more towards machine learning approaches to trying to use that type of information to go back and look at classifiers. I'm not going to talk too much about prediction. I want to move through into the network side of it. But that would represent the sort of flow that we take at the moment with analysis. I should emphasise again that this is really quite robust, unlike what you're going to hear about with metabolomics and the novelty with epigenetics. This has had its dark days, but actually these analytical processes are very robust and really quite easy to access in things like R or various off-the-shelf bits of software. There is a further thing we can start to do with these types of data. When we're looking at this idea of the cascade of the system's function, the avalanche moving downhill, we know that something within this has actually started the process. And we have a variety of causal approaches where we can start to examine whether some effect we see within the level of transcriptomic data is perhaps being caused by another transcript. Now, we can move into, there's obviously a fairly broad swathe I've just cut through approaches. You could move through Markov chain analysis and straight on to Bayesian thinking with that. But actually, you can approach this or start to form the basis of a network idea. The idea which I've ruthlessly lifted from my social scientist colleagues of the transitive triangle. The idea here on the left that if these are people, if A knows B and C, that B is more likely to know C is still actually true in this context. And this can be used with the known literature to start to actually map expectations of whether something, a gene had a high level of transcriptomic expression or a low level of transcriptomic expression. And we can test those expectations back within the data and against the phenotype. This is a very literature-driven type of approach as I'm describing here. What I mean by this is it actually really fundamentally relies on having pre-knowledge. We need to know that one protein affects another and does something else. So somebody needs to have published that. But there's a lot of information there that can be used this. So now, I don't know if anybody are aware of this, but there's a company out there that have produced a package called Ingenuity Pathway Analysis, which does this type of analysis quite effectively. This is their publication here on the right. There are then other approaches to causal analysis. Certainly Ingenuity don't own this type of approach. And there's a number of ways that this can be done within R, and a number of different takes on it that actually move away from the literature-driven side of it. But this is a very useful starting point for this type of analysis. I'm going to shift towards networks within a slide the same. But I'm going to now use this slide to introduce a concept that's been mentioned once or twice, which is this idea that we're talking about the different omic sets one at a time. Actually, we can throw them all in together. And if we do that, and we have this multi-omic type of analysis, we start to get some real benefits on the analysis, on the multivariate side of it. There was a very nice review published on this by some leading names in systems biology a few years ago, and they produced this figure representing some fairly simple stats, which I thought was really quite a nice way of thinking about it. What they're trying to say is if you take like a standard signal distribution, say like a T distribution, and you look at the noise within the data, you can see as the test statistic goes up, that the noise is dropping, and we have a signal window. And very simply, if we start to actually bring multi-omic data together, we squelch the noise and we simply increase, we don't change the ability, the distribution of the T statistic, but we change our ability and our window to be able to pull this. Now this is, I think, quite a nice way of thinking about something that's very hard to explain any other way. If you had two omic sets that were truly independent, you could of course multiply the probabilities together. Of course they're not truly independent. So I think this is a very good way of thinking about it. But what's for sure is that if we actually integrate data, we get a benefit on our ability to, our power even, the ability to detect, and in a way that's actually quite hard to model. It's not, I mean, power calculations are frustrating at the best of times, certainly I find them so. It's actually, I was challenged to say it's impossible to actually model this. All you get is the idea that if you integrate data, you get an increase in power. So, we know an awful lot about how proteins and genes work together. These types of data interactions have been accumulated over quite a number of years now. An early criticism about the type of analysis I'm now going to talk about is it's very dependent on what we know. And of course, this is true, but we actually know quite a lot. So I'll put a perspective on this for you. This is BioGrid, which is a database where we deposit what we know about how two proteins interact. We call this the interactome. A number of years ago, I believe this version of BioGrid was from 2013. There was 14,334 proteins in there with over 65,000 interactions. This next model from last year has gone up to 18,000 proteins with 217,000 interactions. And bearing in mind we have an upper limit of say 23,000 proteins roughly in the human genome, or represent or derive from the human genome, we have a lot of information. So we can consider how our transcriptomic data interacts in a network perspective and set it against what we know we see within these types of data sets. So network analysis allows us to actually go a little bit more freeform on the idea of the causal analysis I was just describing. I don't actually need to have any form of causal information to get involved in this. I need to simply know how things are likely to stick together. I can do that by comparing it with a database like BioGrid that I've just shown, or I can do that via a straightforward correlation network without any assumption as to what the underlying interactions are. This allows me to generate a network of interacting components within the transcriptome that I have. And we can then start to look at the network that we have derived from areas that might imply some level of function. Somebody who stood there flicking that stone downhill to generate the avalanche, where have they stood? And there are several things we know about network structure and network topology that gives us certain clues as to where this may occur. Some simple ideas are like the idea of clusters. A cluster can look a bit messy to define mathematically if you've ever seen some of the ways, there's actually quite a degree of contention as to how you go about doing it. But there's an underlying simplicity. It's where you have more connection within a unit of the network than you have moving outside it. So things that are stuck together tend to do things together. And that still is true within biological networks. And we've already covered the idea of the transitive triangle. That if we can find these cycles within the networks, then that in itself is implying some level of regulation, which gives us certain ideas as to where mechanism may be impacting. It's very important to realise that there is actually quite a lot of information within the biology community relating network structure to biological function. Without the supporting bits of information, it would be very hard to justify what I've just said. You know, we need evidence. And fortunately within biology, we can step away from humans and we can move right down the animal chain back towards things like yeast. And we can go in and we can knock out genes as much as we want without any form of ethics problems. So they were able to take, going back, this paper's from 2007 I think, but actually going back to the early 2000s, a lot of work was done with that type of approach, showing that if you looked at what we knew about the interactome within simple organisms, and you went and you knocked out things sequentially, you got rid of the gene, you could actually see the effect. We could determine some fairly simple things that we'd expect, that regions that were highly connected were much more likely to be functional, that there was a correlation between cluster structure and functionality. And in this work they were looking at the idea of a bottleneck being controlling information, and they were able to actually move towards a statistical summary of that because they were able to go in and sequentially knock out each gene. Of course, in any of the social contexts that we're going to talk about, we can't do that, but we have to have the information to justify what we know about the human data. Where we have been able to look at, and both these papers now are within human patient groups, and we see here this on the left hand side, the classic scale-free network distribution idea, that we have a degree along this bottom axis, and we have logarithmic scale of the frequency, and we have the straight line moving down as classic of a scale-free network. And what see, if we look at genes that are associated with cancer, and genes that are defined here as essential genes, which are genes that we know if we knock out in other animals are fatal, against control genes, we can see a shift of this. So we're seeing a shift of connectivity across the range of connections within the network. And the same with betweenness, which is another measure of network topology. To finish this section, I'm going to come back to a paper that really affected my thinking in the last couple of years about networks, and this is looking at a fundamental question of development, and I think something that really impinges on social sciences from the point of view of how environment and genes interact. We have this idea of developmental differentiation. So we have this chap, Conrad Waddington, came up with this image from quite a long time ago. Conrad Waddington was an English developmental biologist, and he came up with the idea that development's like rolling the marble here through a shaped landscape like this. It moves down, and once it's gone down one of these gullies, it's not coming back again. You're in this, you've made this decision in development, and you're going off down there. There's no return. So the timing and the trajectories you take towards those decisions in development are really quite critical to health, happiness, I would suggest.